Can we start, ma'am?
Give me 5 minutes.
Okay.
Is everybody in? How many participants are expected?
40.
40.
Yes.
Then there are only 18, no? No.
Joining, we are putting the message on group.
So let us wait, no? At least some 60-70% should be there, right?
Okay.
Dr. Jairam?
Yes,
the participants, what is the background?
Participants, they are from assistant professor, and some are from chemistry and some are from pharmacy background. It's a mix.
Some are from pharmacy also?
Also, yeah.
Okay. Let's wait till 9:05 and then we will start.
Okay, ma'am, no issue.
5 minutes at the most. That's it. Just for any internet issue, we will wait and then we will start. I think we can start now.
Okay, ma'am. Arundhati.
Yes, sir. Good morning to one and all, and welcome to the first session for today. It's a great honor for me to introduce you all to our eminent speaker for this session, Dr. Padma Devarajan, who will be taking this session on drug delivery systems for cancer. Just a second. Dr. Padma Devarajan is a professor in pharmacy and former head Department of Pharmaceutical Sciences and Technology at the Institute of Chemical Technology, Mumbai, India. She is a member of the Board of Governors, President of the Innovation Council, and coordinator of the World Bank Technical Education Quality Improvement Program, or TAKIP, at ICT. Her research focus is in drug delivery systems and diagnostics for human and veterinary healthcare. She has filed many patents and licensed patents to industry, of which two products are commercialized. She has edited and authored two books on targeted drug delivery published by Springer. She is a recipient of several national and international awards and several innovation awards. Her latest award is the Punjab Rao Deshmukh Outstanding Woman Scientist Award 2019, conferred by the Indian Council of Agricultural Research, Government of India. She is also the immediate past president and first woman president of the UDCT ICT Alumni Association. She has filed over 20 patents internationally and nationally, has 7 patents granted and 4 patents licensed. Her research is funded through a number of grants from the Government of India, including the Department of Biotechnology, Department of Science and Technology, Department of Atomic Energy, Council of Scientific and Industrial Research, Indian Council of Medical Research, University Grants Commission, and the All India Council for Technical Education, and for the industry, including companies from Japan, Germany, and USA, and is a consultant to the pharma industry as well. We welcome you, ma'am. Over to you now.
Thank you, Dr. Arundhati. And let me first start by thanking Dr. Golave and the management of SPCE for this very kind invitation. I am used to talking to pharma crowds a lot, students, faculty. This is going to be slightly different. And this is something that I have been doing, talking across engineering colleges in the past, I think 1.5 years, about different aspects, including innovation and the innovation and startup policy. But I think this is the first time I am going to talk about a very biological, a very scientific topic, which is non engineering, to a group of scientists, particularly faculty. So I have tried to structure the topic in a way that, anybody with a basic science degree would be able to catch up and would actually get some understanding of what I'm trying to tell you. So with this, I think I will like to start sharing my presentation. So just give me a moment. Is my slide visible?
Yes, ma'am, it is visible, but it is not in presentation mode.
Oh, God.
I think you have to share initially entire screen, so that will work out.
Let me just try once.
Yeah.
Yeah,
Entire
I'll do that.
entire
Just give
screen
me a
you do.
moment.
Yeah.
I'll just stop sharing and again share.
Again you share and select entire screen. That will be better. That will work.
Okay, stop sharing. But so now I wouldn't know how to get to my Just give me a moment, please. I'll
Yeah,
get back.
you do entire screen or entire
Now,
window.
can you see my entire screen? I'm
Your
sharing
screen
my
is there, yeah,
Ah,
but
but now here I don't know how to find my file. That's what I'm trying to do. 1 minute. It doesn't seem to be selecting anything. I think I'll just stop sharing. Give me a moment. I'll
yeah,
just see what
yeah,
I can
yeah.
do next.
From beginning you can select, madam. From beginning, slideshow, and then from beginning.
Now, could you see it in the slideshow mode? Yeah, now is it in the slideshow mode? They can't
No,
even
ma'am.
hear me.
No, ma'am. Not it.
Not yet.
Yeah. You go in slideshow and then click from beginning. Maybe. Yeah, yeah. This icon also will work. Yeah.
Yes. So now I'm pressing slideshow.
Okay. So that is the problem, ma'am. Actually, when you are sharing na, the
Huh?
three options will come.
Huh?
First option is your entire screen. Second
Huh?
option is a window, and third option is a tab. You have to
Okay.
select your entire screen, so that will work.
Okay.
Yeah, yeah,
Yeah. Okay.
yeah.
Now?
Yes, ma'am. It's perfect now. Done. Good.
Ah.
Yeah, yeah,
Okay.
yeah.
Thank you.
Yes,
Thank you
yes.
so much. So
It
this,
happens. Yeah, when the platform is new, it creates like
yeah.
some
No, the
confusion.
platform is not new. My laptop is new.
Oh, yeah. But now it's
That's
perfect, ma'am. You can
No, so
continue.
this actually talks of collaboration between engineers and biologists.
Yeah, yeah, great. You capture perfectly.
Yes. So
Okay,
sorry
okay.
for this small
No issues, ma'am.
technical
It
glitch.
has been
Yes.
on the set now. You can
Yes,
continue.
yes, yes.
Yeah.
So, very good morning to all of you. And as you have already been told, I'm going to talk about anti cancer drug delivery. And it's a huge area. So, what I'm going to present to you is an overview of the science and the technology. Because cancer, we know, is rampant and it is supposed to be the first or the second leading cause of death across the world. And although it is seen that people up to 70 are more prone to cancer, what we find with longevity increasing, that people 75 plus, 80, 85, the last stages of last leg of life, if we may say, are also being detected at that stage with cancer and suffering. So cancer has become a disease that needs to be really tackled, and there's a lot of work that's going on in cancer. So I have structured the presentation in a way that it would be more like a popular science lecture, so that anybody with a basic science background would be able to follow what I'm trying to say. There would be a little involved biology, but not too much. So I would be presenting to you cancer, an overview, and what is metastasis, which is one of the biggest challenges of cancer. The second aspect I would be presenting to you is the tumor microenvironment, because we all know when we talk of cancer, the one word that all of us think of is a tumor. So I would be presenting to you certain relevant, important aspects of the tumor microenvironment, which are important to understand the process of drug delivery in cancer. Then we would move on to what is the major stronghold for cancer therapy today, and that is nanomedicine, which originates from nanotechnology. So a brief about nanotechnology and nanomedicine. And then we move on to the next step, where we focus on nanomedicine in targeted drug delivery, because nanomedicine has other applications also, which I will not be touching. And we will be looking at nanocarriers, which are the basic intervention in nanomedicine, which is used to finally deliver the drug for therapy. So let us begin with cancer. So according to the WHO, cancer, like I mentioned already, is the first or second leading cause of death before the age of 70 years, a huge concern, and the spread of cancer is growing so much that I'm sure today each of us knows one or maybe more than one cancer patient. Earlier, it was not like that. Cancer was something for others, not for us. But today, it's become an issue that could hit any of us at any point of time. So when I use the word cancer, what are we talking about, or what does it imply? So the hallmarks of cancer, which is also termed as malignancy, as opposed to what is benign growth. So malignancy is growth, which is cancerous. You also have today people come who have different types of growth. They are worried whether it's cancer, and then it turns out to be benign. So what is this? There is growth there, and there is growth in cancer. You call one malignant, you call the other benign. So when is it considered malignant? When the cancer cell, so it's called a cancer cell, when it exhibits independence from growth signaling. So it stops listening to the body. It stops listening to the body signals. And therefore, it continues to multiply without limit. Otherwise, the body would tell a particular cell to multiply at a certain rate. For instance, our hair and our nails, they grow faster than other cells. But we don't have our stomach and liver cells just growing indefinitely. So when you have this independence from growth signaling, which are signals that stop and rather control, there is no limit on proliferation. Now, what is also important is when there is proliferation, the body tries to kill those cells and terminate them, but these cells can evade apoptosis. Apoptosis is cell death. So they cannot be killed easily by the body mechanisms. What they also develop is angiogenic ability. Now, what is angiogenic ability? It is development of new blood vessels to allow them to grow. And then what is equally important is it's not that you develop a cancer at one site, say a breast cancer or a liver cancer or a brain cancer. From there, it can invade. So invasion and metastasis, which means moving across. Metastasis means going to another place conquering that place and then killing those cells also. So creating a new tumor there. And insensitivity to growth inhibitory signals. So these are hallmarks of malignancy. So when you have any of these or one or more of these, you would consider that as a malignant condition or a cancerous state. You have various types of cancers depending on which part of the body is affected. For instance, if it's the epithelium, it's called carcinoma. If it's muscle or connective tissue, you call it sarcoma. If it's a gland, you would call it a carcinoma of that gland, for instance, adenocarcinoma. And if it's the bone marrow, it's called myeloma. If it's a blood cell, you call it leukemia. If it is the embryonic tissue of organs, you call it blastoma. If it's a lymph tissue, it is lymphoma, and then you have mixed cancers. But all of them have one common feature, and that is uncontrolled cell division, which results in the formation of tumors. And these tumors may be solid, may be liquid also, and when they are liquid tumors, they are even more difficult to treat those cancers. But if it's a solid tumor, today medicine has advanced significantly, and it is possible if a tumor is completely removed for 100% remission and the person does not show another episode of cancer. So when we talk of tumor, this is just a picture for you to visualize something. So you have a blood vessel and tumors would need circulation, which is very important. And when you have such cancerous cells, here you have one cell that is depicted, and this is what would be attacked by the drug delivery system or by the drug. Now, what we also need to understand is tumors can grow up to about 2 mm, but beyond that, they need blood vessels. And that's why we looked at anti-angiogenic factors they produce, and they have pro-angiogenic factors produced to create blood vessels so that the entire tumor, as it moves away from the central blood vessel, can also be perfused with blood. So this is a picture that is showing you how a tumor has grown so big, and it cannot grow further unless you have blood vessels coming up. And then look at this, how blood vessels keep growing to help the tumor to keep growing further and further. So these are all specific features of cancer cells because they ensure their growth. Now, you have tumors formed, but the biggest challenge is not just the tumor, but when there is metastasis or a metastatic process ensues. So this is an example, a kind of video which I'm showing to you on how breast cancer is known to metastasize. And many times a breast cancer patient does not die of breast cancer, but dies finally of brain cancer or lung cancer or liver cancer. So what happens is you have seed cells from the tumor that circulate. So how does that happen? Watch. So you have the seed cell, it goes through the blood vessel, and then at some point it starts extravasating and then forms the metastasis. So this is a tumor that is formed, and once the tumor is formed, you have angiogenesis that is triggered, and this then helps in proliferation and the tumor grows even more. So this results in what? It results in the cancer spreading to different major organs, maybe the lung, the liver, or even the bone marrow. So it gets arrested in host organs and then it develops there. So it's like typical invasion, like you had the Mughal Empire, maybe if we take an illustration, coming into the country and occupying different places and then making those places their own. That's what the cancer cell tries to do. So metastasis is still one of the biggest challenges in cancer therapy. So that was one aspect that is very important to understand about cancer, that even if it starts at one place, it has this tendency to go to other places. Now, what is important in cancer therapy is also to understand the tumor microenvironment, because that's where I want the drug to reach. So I am just taking certain highlights, okay? So one is normal vasculature versus tumor vasculature. So if you look at normal vasculature, you find that it is very continuous. It's very continuous, there are no breaks. But if you look at the tumor vasculature, can you see here there's a break, here there is a blind end, there's a break in the vessel wall, there is a shunting between the artery and the vein, which normally would not happen in normal cells. So it's going to get all mixed up, the blood. So basically, you can look at this, which says they're highly irregular, they're tortuous. Can you see how they bend, whereas these are relatively more linear? They have arteriovenous shunts like this, blind ends, lack smooth muscle or innervation, and they also lack endothelial linings and basement membranes. So which means from this region or to this region, there is scope for material to go in and out, both ways. So when in, you use it to advantage by sending your drug in. Out could be the cause for metastasis. What is the other way important characteristic of a tumor? It is hypoxia. And hypoxia increases as the tumor size increases. So as the distance from the nutritive blood vessel increases, hypoxia increases. So if you look at this, here you are looking at something that's quite close. So you can have a lot of drug going to the smaller tumor, but as the tumor becomes larger, you have hypoxic cells developing. You have hypoxic cells developing. These hypoxic cells develop, on one hand, drug resistance. So they don't allow the drug to go in, and they develop drug resistance, and they also develop metastatic potentiality, which means they can leave because they are getting choked. So they want to leave from there. So they try to move out of the tumor, and that's how metastasis could occur. So that again is a very important aspect to understand, that it's not very easy to deliver inside a large tumor. And then what else is different from the normal tissue? That is a pH. So as you move away from the nutritive vessel, you have an tumor interstitial pH of about 6.5 to 6.8. And as you go into the cell, it goes further down 5 to 6. And if you go into the lysozyme of the cell, or some phagosome of the cell, or an endosome of the cell, depending on the nature of the cell, the pH can go even lower. So pH is again something that is very important, that one needs to understand. And actually, you exploit this to deliver to the tumor. So you'll have something that will not release here, but will release at this pH when it gets it. So pH is very important. And here is a picture, an image taken of a tumor, an actual tumor. And this tells you pH for the blue region is 6. So here you can see the tumor pH is not 7.4, which is the blood pH. It is significantly lower. So that was a real brief on cancer, understanding the tumor microenvironment. And all this we will now use as we move along to talk of anti-cancer drug delivery. So we move on to the next aspect that I said I would talk about, and that is nanotechnology and nanomedicine. So what is nanotechnology? It is the branch of technology that deals with dimensions and tolerance of less than 100 nanometer. That's how nanotechnology came in a big way, saying it has to be less than 100 nanometers. And related to nanotechnology, which has immense applications in diverse fields, one of the major fields where nanotechnology has application is nanomedicine. And this is the application of nanotechnology in therapy, and that is what we call as nano drug delivery. And importantly today, nanomedicine is also having a lot of applications in diagnosis. And you also have a combination of therapy and diagnosis and a new branch that has emerged in nanomedicine called theranostics. One major application in nanomedicine is anti-cancer therapy, which is the only application I will be discussing today. So now I would like to take you first to this 100 nanometer size. Nanomedicine has kind of redefined this 100 nanometers, because in nanomedicine, it is seen that if you have less than 100 nanometer, it's very good. But you need technology to create particles of that size and to create them reproducibly and to ensure that they are stable till they are used. So from the formulator or the manufacturer to the user, you need stability. So when you're talking of stability, you must understand that it is easier to stabilize particles greater than 100 nanometer and more difficult to stabilize what is less than 100 nanometer. So if you look at nanomedicine, you will find lot of nanosystems of 100 nanometer and less, but for different applications in nanomedicine and even for cancer, there are methods developed where you could even use 200 nanometer size nanoparticles. And for other applications where nanomedicine is becoming important, and that is infectious diseases, and one of the biggest and important drug in the nanomedicine form for infectious diseases today is amphotericin B to tackle mucormycosis, one of the biggest side effects of COVID therapy. There, you can go to larger sizes for infectious diseases, and there's a lot of work our group does on infectious diseases and also cancer, but we work on small size for cancer and we work on larger size. So typically for nanomedicine, I put that question mark there to tell you that size is not limiting. You can go even up to 300, 400 nanometer size depending on the application. So coming back to cancer therapy, what are the challenges? Now, the biggest challenge is the treatment. And this treatment many times is such that the person recovers from cancer but suffers the side effects. The side effects of conventional cancer therapy sometimes are terrible. Today, you have monoclonal antibodies that have come in, and they have reduced the side effects significantly. But the cost is so high, it's not affordable therapy. So a common man cannot afford that kind of therapy and yet has to suffer these kind of side effects. So that is one huge challenge. The other is post-treatment. Can somebody become normal? Now today, medicine has advanced and most people are normal, but there is still a percentage of patients who almost become crippled after cancer, may not always be walking on crutches, but they could have different kind of side effects that makes life really unbearable. So in cancer therapy, it's very important to address this aspect, and that is quality of life post-therapy. The other challenge in cancer therapy, particularly using nano, is big fears about the science of timing. Now, this is the inside of a cell. This is the DNA. This represents nanomedicine. The people here represent nanomedicine. And these are all the cell organelles. So imagine this child is suddenly let loose inside like a garden, and what's going to happen? He or she could destroy anything here. And that is one challenge of nanomedicine, where one is not sure whether these kind of destructions can occur, because what enters the cell first is nano, and it enters in larger concentration than the conventional drug. So both ways, whether there could be serious toxicities, it's all being studied and explored, and you still, after all that, you have commercial products in the market, although small in number. So with these challenges in cancer, and of course the challenge if you use nano in cancer about entering the cell, nanotechnology plays a very important role today in targeted drug delivery. So now when we talked of cancer, what were we talking about? We were talking about side effects. And why do you have side effects? Because you're delivering the drug to the tumor. Simultaneously, you're delivering the drug also to other parts of the body where it is toxic. So I need to limit the drug delivered to the other parts of the body, so that we call that a systemic delivery. And I need to focus on, let's say, tumor delivery. So that is what one tries to achieve by targeted drug delivery. And targeted drug delivery is possible because of nanomedicine. So what we are now going to look at is nanotechnology in targeted drug delivery, the relevance, the applications, and more details. First, we will try to understand where this entire concept originated. Now, the picture you see here is Professor Paul Ehrlich, who way back in 1898 suggested that bodies which possessed a particular affinity for a certain organ could be used as a carrier to bring therapeutically active groups to the organ. So this whole concept that you could have a magic bullet by taking your drug with some molecule that has an affinity, say a molecule that has high affinity for the liver. So you attach that molecule to the drug and you would have all the drug going to the liver. So that was just the first thought process. It has not been simple, and that's why we see that more than 100 or how many years later, I don't know, we are still not reached a very ideal situation where you say every cancer we have a medicine, we can treat it without toxicity, etc. But this is where it originated, and over the past century, a lot of work has been going on, and what we see today is what originated as a thought in 1898. And then, of course, you had Feynman, who brought in the concept of nano, and both put together is what has emerged today in the form of nanomedicine-based targeted drug delivery systems. So what is the advantage of this targeted drug delivery system? Increased efficiency, reduced side effects, because you're talking of systems that would go more to the cancer. And then because of that, you're going to have higher concentration in the tumor, so you can reduce the dose, reduce the number of doses, and therefore get better patient compliance. Because if there are too many side effects, sometimes patients say, I'll suffer the disease, I don't want the drug. But is it so simple? Is it like taking a gun, you have a round circle and you start shooting like this, what you see here? It's not so easy. The body has its own protection. It has a lot of defense systems. So these physiological barriers refer to the defense systems of the body that need to be overcome. After overcoming these barriers, you need to deliver the drug to the site of action. You deliver it, and it should be there for some period of time till it can actually show effect. And when I deliver it to the site, I must also assure safety. So if I need only 10 and then I am delivering 50, it could cause toxicity. And if I need 10, I'm delivering only 5, that may not be enough. So I need to deliver adequate amount, maintain for adequate time, assure safety, and all this after crossing physiological barriers in the body. So based on targeting, one normally talks of different levels of targeting. And when we talk of level, it is in terms of ease. So primary is directly to an organ. And if you see here, it's the liver or the brain. The secondary is cellular targeting. So say within the liver, in the liver, we know we have different types of cells. And I'm sure anybody with a science background would have heard of Kupffer cells in the liver. Kupffer cells represent the defense cells in the liver. And then you have the other cells, major of which are the hepatocytes. So I can take my system to the liver and deliver it only to Kupffer cell, or I can deliver it to hepatocytes and bypass Kupffer cells. That is secondary targeting, which is more advanced. And then I have tertiary targeting, where I deliver it into the cell, and inside the cell, I can direct it to the nucleus, to the mitochondria, to the lysozyme by having different kinds of ligands. So if you look at this picture, it only tells you how easy it is. So primary targeting is like, it's not like walking on a road, certainly. Targeting is not very simple. So it's not like walking. You have to put in an effort there. So it's like jumping, which is not very tough. When you come to secondary targeting, the degree of difficulty increases. And when you come to tertiary targeting, it's even more difficult. So what one needs to understand is one has to identify what is an appropriate type of targeting and then decide where one could focus to get the best efficacy and least toxicity. So when you talk of primary targeting, you can deliver to that organ easily, like I said, liver or brain, but you have to ensure that it is not thrown out by the organ quickly. In secondary targeting, both delivery and retention is a challenge because delivery becomes more difficult. And in tertiary targeting, you may deliver, but bioavailability is a challenge, which means, let me say, I want to deliver into the endosome, and my endosome pH is about 5 to 6, and at that pH, my drug degrades. So what do? I have reached, but it's not going to be efficacious. So every aspect needs to be looked at very carefully. And understanding these different levels of targeting, how exactly am I going to target is what we're looking at now. How exactly am I going to target? So you have passive targeting and you have active targeting. The passive targeting and you have active targeting. What is passive targeting? You can see this boat bobbing up and down. Is the boat doing anything? No. It's the waves that is taking the boat in its direction. The waves determine the direction of movement. So in passive targeting, what you do is you inject the system in the body and allow the body to take it. So wherever the body decides to take it, it would go. In active targeting, it's like using more control. You have a torpedo and you can define the site, right? So you can direct this 10 kilometers away. It should fall at X point and it would happen. So that's active targeting. So when we are looking at targeting, we can have both passive targeting and active targeting. And in case of passive targeting, there are different approaches that we can follow, active targeting, different approaches. But what is one fundamental principle there? The fundamental principle in enabling passive or active targeting is to understand the body physiology. Because we're going to inject the system into the body, and we need to understand whether when I inject, what's going to happen to my system? If I give it orally, what's going to happen to my system? And all this is known. So physiology is already understood. So once you understand body anatomy, body physiology, you can exploit appropriately to decide whether you want passive targeting, whether you want active targeting. So what are the opportunities? So when you look at active targeting torpedoing, it's a little more difficult. But passive targeting is easier in terms of development of the system. Okay. So when you're talking of targeting, we also need to address the technology. That's why I said overview of science and technology. Because I may say it's good, I can use this X ligand and enter into a cell, etc. But the question is the ligand available? How do I attach the ligand? So a lot of things. So whatever decisions one is taking about targeting needs to be in the light of understanding the science, the physiology of the body, and the technological feasibility. So what we are seeing here is what are the possibilities for passive targeting. So I already told you you have primary, secondary, and tertiary. So let us look at primary, which is the simplest form of targeting. So this may not be too relevant for cancer, but this, yes. If there is a cancer in the eye, you could look at an ophthalmic injection directly into the eye. There are intravitreal injections and other injections given in the eye. So you could have a system designed that way. If it's pulmonary, you may look at something inhaled directly into the lungs. So that would be anatomical targeting. And what you see here is injecting into an artery to go directly into a tumor. Secondary targeting is where you need to address the body physiology. And this reticuloendothelial system is the major defense system in the body. And what is the role of this reticuloendothelial system? The role is to engulf and destroy foreign substances, bacteria, viruses, etc., like you see in this video here. So this animation is actually showing you how, when there is foreign invasion, the cell gears up, phagocytosis, and destroys it. And it's because of this mechanism that is continuously happening that today all of us who are here are well, fine, and not under attack. But when you're sitting in the same place, suddenly you will get up and then one person falls sick the next day because their defense system is not as good. So all of us are talking today of building up immunity, which is very, very important. Now, which are the major organs that represent this defense system? It's the liver, the spleen, the lungs, and the bone marrow. The liver, the spleen, the lungs, and the bone marrow are the major defense organs. So now what are we watching? We are watching how the reticuloendothelial system is capturing foreign substances. And if you see, they are particulate, right? Bacteria, viruses. So if I want to deliver into the system, into the liver, spleen, lungs, bone marrow, I can use this approach of sending particles which would get taken up. But if I don't want to go into the reticuloendothelial system, so let me say I have the liver and I have the spleen, and I want to go outside, I don't want to go into the RES, then I have to have a method by which I can bypass this reticuloendothelial system and reach my target. So when I'm talking about passive targeting, I'm talking about two aspects. One is going to the reticuloendothelial system, which is very easy. And the other is going outside. Now, when would I go into the reticuloendothelial system? When I am looking at treating infectious diseases, particularly intracellular infections. And when I'm treating something that's not an infection like a cancer, I need to go outside. So I need to bypass the reticuloendothelial system. And here you will see a word stealth. So I need to hide and go past. That's what stealth means, to hide. So I need to hide and I need to go past. So there are strategies that are adopted to hide, which we will also be looking at. So basically, we're talking of now passive targeting, either to the reticuloendothelial system or outside the reticuloendothelial system. Now, one more approach is passive targeting, where you exploit the pathophysiological condition. It's called pathophysiological passive targeting. Now, when we talked of the tumor environment, we told you the blood vessels are broken, the blood vessels are more tortuous, the gaps between blood vessels are more. So what you're seeing here is normal vasculature, where the endothelium is very closely knit. And this gap is generally just about 20 nanometers. But when you go to tumor vasculature, gaps start increasing and it could go up to even 700 nanometer. This is where your tumor has grown. Okay, it has invaded and it has grown. So these gaps will allow your nanocarrier to enter. So now when I click, you will see that how these particles move and how these particles move. Please watch. So they just move straight. They cannot leave that blood vessel. But if you look here, particle 1, 2, 3 went into the tumor, 4 also went in, 5 went in, but 6 could not go in, it came straight, 7 came straight. So this is just a kind of schematic that we're trying to show here. So what we're trying to say here is, here there is a possibility for these particles to take advantage of this fenestrated tumor vasculature and cause enhanced permeability and also retention. Once it enters, normally it doesn't come out easily because the tumor gets more blood vessels, but it is not able to encourage lymphatic circulation through which you have drainage that goes out. Okay, now what is the other aspect that you can look at for passive targeting? We can look at another very important aspect, and that is size of the particle. So when I look at size, I would like you to look at this. This is micron, so this is about 8 microns. So RES uptake of particles happens when the particles are big. But if you look at this extravascular tumors, if the size is less than 100 nanometer, it can go to extravascular tumors. And why is this? Because less than 100 nanometer is generally not recognized by the RES. Less than 100 nanometer. So I can use that advantage and go to less than 100 nanometer size to bypass the RES and go to a cancerous tumor. And then when I go to the site of the tumor, I take advantage of this extravasation. I take advantage of the distorted vasculature in the tumor to allow my particles to go in. So I can use size to my advantage, coupled with the enhanced permeability and retention effect. So that is again an approach of passive targeting. And what did we say passive targeting is? You inject and allow the body to do. So these particles would keep circulating and they would be moving the way the body takes it. So when it comes here, again and again, there is a good chance that more and more particle would keep accumulating with time in the tumor. But you cannot have a method where it would come here and then selectively go in. Whether it goes in or not is not predictable, but the chance, the possibility that it can enter becomes very high. And if it is less than 100 nanometer, the possibility of it coming back to the tumor site becomes very high, and therefore more and more can accumulate. The next approach, which I told you, is active targeting. And active targeting involves lot of stimuli. So one is, I needed to hide from the RES. Okay, that's very important. And then I can use different stimuli. I can use receptor mediated targeting because a lot of cancer cells overexpress receptors. For example, folate receptor, transferrin receptor, ursialoglycoprotein receptor, adhesion receptors, and there's a whole list. VEGF receptors or mannose receptors, scavenger receptors, and it goes on. So if I have some ligand that can attach to this receptor and I attach that ligand to my nanoparticle, it can preferentially attach to those cells, those cancerous cells, and that's how I get active targeting. So folate receptor, a simple ligand is folic acid. Transferrin receptor, a simple ligand is transferrin. ASGPR receptor, a simple ligand is galactose. So you do have simple ligands and you have very complex ligands also. But I can use that approach. I can use antibodies to target antigens overexpressed on cancer cells. And then I can use stimuli. What are these stimuli? I can have tumor microenvironment triggered because we saw pH in tumors is lower than plasma pH, but temperature in tumors is generally higher because of rapid metabolism. So you can exploit temperature changes, you can exploit pH changes, or you can exploit both together. Or you can totally use triggers which are external. So you can heat from outside if the tumor is quite superficial. You can use magnetic energy or ultrasound energy to trigger release by active targeting. So the system would keep circulating. When it comes to that site where you have applied a magnet, it would release the drug. So that's how you can target. So in case of receptor mediated targeting, just as an illustration, you need to find out which is the overexpressed receptor. You need to find out which is the ligand, conjugate the ligand to the drug-loaded carrier. And once you've conjugated, this black is a ligand, it would attach and then it would be taken up by a process of endocytosis. If not, it could be phagocytosis if it's a macrophage or a phagocyte. But a general cell, it would be endocytosis. And endocytosis also, there are different mechanisms. The most common is clathrin-mediated endocytosis. But various receptors, I already told you these names, you can just quickly see. For instance, like I told you, mannose also, EGFR, VEGF, also LDL receptors. A lot of receptors can be targeted. And then we come to the tertiary targeting, where you can have the lysozyme being targeted, the mitochondria, the endoplasmic reticulum, or the nucleus in the various cells. So with this basic introduction, because I thought without that, if I only spoke to you about the carriers in drug delivery, you would not be able to relate it to cancer. So we've had a brief overview on cancer, the tumor microenvironment, the concept of targeted delivery using nanocarriers, and now we will look at the nanocarriers. So this magic bullet concept was proposed by Paul Ehrlich in 1898. But today, you call it as a magic wand, where you don't have just the drug and targeting moiety the way Paul Ehrlich proposed, but you also have a nanocarrier. So this nanocarrier has come in through the concept of nanomedicine. So what would be an ideal nanocarrier? It should be biodegradable, which means once I inject, it should degrade in the body to save substances and be eliminated. It should give me high target specificity. It should give me no toxicity. So these are important aspects on the biological front. Now, again, I may have the best nanocarriers here, but if I cannot exploit it technologically, it is of no relevance to society. And we know cancer is a problem that is growing in society. So if I have just something theoretical, it's not enough. I need something practical. So I need to look at high drug loading, which means there should be good enough drug loading, commercial feasibility, and affordability. So commercial feasibility would talk of the technology, the scale up, the reproducibility, etc. And then finally affordability, because today you do have anti-cancer therapy, which is targeted, but it's not affordable. So it's important to work in that direction to make it affordable. So how would you administer these? I showed you primary targeting where you could go to pulmonary intratumoral. This is very, very limited. Okay, very limited, does not have major applications. The major application is parenteral by the intravenous route. And today there is focus on trying to use the oral and other routes for targeting. For instance, our lab has been doing some work on orally delivered anti-tubercular nanocarriers, tuberculosis and cancer, which can go in much higher concentration to the lung, so for lung targeting. So it is possible to exploit the oral route also. And very recently, I haven't put it here, but we've been able to target the bone by sublingual delivery, that is delivering below the tongue. And we looked at salmon calcitonin, a protein, because that cannot be given orally because it would be digested in the GI tract. We gave it sublingually using nanoparticles, and we found very good data on bone targeting. So these are the routes. Now, why does one look at nanocarriers? Enhanced drug absorption, improved bioavailability. They can protect drug from degradation, and most important for cancer, targeted delivery and reduced non-related organ toxicity. Very, very important aspect. So what are the pharmaceutical nanocarriers? Here you see the size. Like I told you, we go up to even 400, 500 nanometers, and that's what's depicted here. So you can have dendrimers, you can have micelles, neosomes, nanoparticles, nanocapsules. Then lipid-based, you have liposomes, you have solid lipid nanoparticles, tubosomes, and then other carbon-based carriers also you can have. So you have a whole range, which we will quickly look through. So you have polymer-based nanocarriers, like nanospheres or nanocapsules. So capsule will be vesicular with a shell and an open pocket inside. Dendrimers, for those who are from the chemistry background, they're also called as star polymers. And dendrimers come in different generations, 1, 2, G1, G2, G5, and I think the latest is G7. And lot of scope to attach a lot of things. So I can attach my drug, I can attach maybe 1 or 2 ligands and get the best kind of targeting. Then I have solid lipid nanoparticles made from lipids. You have what are known as nanostructured lipid carriers, which are made from lipids which are solid and lipids which are liquid. And it is seen that when I have this mixed lipid carrier, it is able to hold more drug for longer time. So I get better stability. And then this is a carrier that we devise in our lab where we combine a lipid with a polymer and have a lot of advantages because the polymer helps you to put hydrophilic drugs inside a lipid carrier. Now, why do I need a lipid carrier? A lipid carrier is very useful when I want to target the reticuloendothelial system, because lipids are hydrophobic and the RAS can rapidly take up hydrophobic. But if I do not want to go into RAS, I need to look at hydrophilic carriers and not lipidic carriers. Then I have the liposomes. These are made from phospholipids and they form spontaneously when phospholipids are dispersed in aqueous environment. And you have so many types. So basically, what are liposomes? They are bilayers of phospholipids with the hydrophobic region inside and the hydrophilic region outside, and they spontaneously form vesicles. So this is a typical unilamellar vesicle formed by phospholipid bilayers. But you could have different types of liposomes. You could have multilamellar, then you could have lamellar, multilamellar, in many unilamellar in one big vesicle. So depending on how you prepare it, and this is a schematic of the liposome. And what you see here, all these spots, is a drug that has been entrapped. So it has an aqueous core and it has a hydrophobic phospholipid bilayer here. So you can put hydrophobic drugs or hydrophilic drugs in liposomes. And liposome, because they are made from phospholipids, are till date the safest nanocarriers. And if you look at the market, most of the nanocarriers that are commercialized today, although the number is not very high, are liposomal. Then you have microemulsions and nanoemulsions. So microemulsions have oil, water, and surfactant with a co-surfactant. The nanoemulsion does not have the co-surfactant. So these are very, very stable systems. These are not as stable. They are not very kinetically stable systems. And then polymeric micelles made from different types of block copolymers. And why block copolymers? Because they have a very low CMC. So at low concentrations, they can form micelles. So that is the reason why block copolymers are used for micellar drug delivery. Now, what I would like to show you are some examples. And mainly, these examples are using passive targeting, and then I'll show you some using active targeting. So in passive targeting, one very important strategy is stealth, particularly for cancer, where I want to bypass the RES, right? I said RES is a defense system. For cancer, I have to bypass the defense system. So the strategy that you use is stealth. And using this strategy, it has been possible to alter the pharmacokinetics, which means the residence time of the system in the blood or plasma. So like if I inject the plain drug, say doxorubicin, and it goes out in say 15 minutes, it becomes half the concentration. If I inject liposomes of doxorubicin, it may remain there for 5 to 6 hours. So I alter PK. So when I alter PK, what happens? I am able to bring the nanoparticle again and again, because it's in circulation, to the site of the tumor and allow more and more to accumulate. Once I do that, automatically safety is taken care of when I try to target. So what is stealth? It's a secret. It means hidden act and a method of escape, like I said, from the RES. And the stealth approach involves modification to increase circulation half-life. That is what I just explained to you. Doxorubicin in solution versus liposomal doxorubicin. Now, when I have the reticuloendothelial system, what you're seeing here is the same picture I showed you earlier, the same animation. But what you're seeing here is a live picture of exactly how this phagocytosis occurs. So this is the actin film forming over the particle. This is a nanoparticle. It's exactly same manner in which bacteria, viruses, etc. are engulfed. And this is that one liposome. If I go back, you will see here. Now this is a plain liposome. This is a liposome that has this. This yellow is stealth, is indicative of a stealth molecule. So in the absence of stealth, see it goes into the RES. But the minute I add a stealth feature, it can go and target outside the RES. So that is how stealth actually helps you to bypass. So now how do I confer the stealth? I'm saying stealth, what is stealth? So advantage of stealth, yes, we've already seen it, altered PK, decreased RES, prolonged circulation time, enhanced this. So stealth is imparted by using stealth agents that can simply be adsorbed on the carrier, or you can even covalently bond. So you have many stealth approaches. We are not going to discuss all. The most common is surface modification. The next is size. A third is shape. And then you have others like mechanical properties, recognition of self, etc. But all this relies on understanding the body defense mechanisms, right? So now, one of the materials very commonly used for stealth, which you should know, is polyethylene glycol. It's a very common chemical. It's used widely in pharmaceuticals across formulations like oral, topical, ophthalmic, etc. What does it do? It's a non-ionic, flexible, hydrophilic polymer. So note this term hydrophilic. I told you lipidic go to RES. So when I coat my particle with a hydrophilic polymer, it causes a steric barrier and prevents opsonization or recognition by the RES. So surface modification using polyethylene glycol results in the particle being hidden from RES because that hydrophilic coating of PG results in the body sensing it as water and not as a foreign material. And here is the example that I mentioned. See the T half here. It's coming to half the concentration in about an hour, say 2.5 or 2 to 1 in less than an hour. But here you see 50 to 80 hours you have this in circulation, which gives you very high efficacy. So pegylated liposomal doxorubicin is already a marketed product. So this is altering the pharmacokinetics or the plasma half-life. Now, the first stealth products based on pegylation were Adagen and Oncaspar, and both these are based on the theory of Paul Ehrlich, where you had the drug directly conjugated to PG. This is not a nanoparticle. So this you can see also early 90s, and it's after that, after 95, 96, that liposomal formulations came in a big way. This is another pegylated product, pelfilgrastim. Now I'm trying to show you some data on how you actually see accumulation. I showed you a very schematic picture where those red particles went, some of them into the tumor, some did not. Look at these two pictures. This is the left side is a normal. It's a mouse, I think, mice. And this is a mice with a tumor here. In this T is tumor. So this is a normal mice, but here you have the tumor, and you see here a good accumulation of the radioactivity, saying that when there is a tumor, there is scope for the particles to go in there and accumulate because of the disturbed vasculature. So what we studied in theory, we're actually seeing, and this is again a similar data that you're seeing, where the tumor is here, and with time, see 4 hours you see so much here, and then at 16 hours you see so much more accumulation. Whereas in the non-tumorous mice, you don't see much accumulation. So these are the tumorous mice where you are seeing accumulation. So this is actually examples of passive targeting. And the drug here is paclitaxel, a known anti-cancer drug. Another method by which one actually sees is by actually sacrificing the animal and looking at the tumor size. So a quick look at the top two rows. This is normal mice not treated with any drug. And then as I treat with drugs, you see here, the size keeps going down. So it will not go down unless drug is accumulating. This is another drug, hydroxycamptothecin. And the same thing can be done not only with scintigraphic imaging, but with PET MRI imaging also, where you see that in case of, now if you look at this, it is talking to you about when it's lighter, there is more of this intensity of the imaging, saying that there is more accumulation there. And this picture also shows you much more accumulation. So two things here. One is that it's possible to use stealth and do passive targeting. And how does one monitor? Using different imaging techniques or actual sacrifice and monitoring techniques where you monitor the tumor size. But we talked of polyethylene glycol, and polyethylene glycol based products are there in the market. But there is a huge issue with pegylation. And what is that pegylation issue? It is a problem called PPES. When you look at this hand, you don't feel it's very bad. But these fingers have lost their strength. So they bend like a glove. They do not have the capacity to hold. So the person who has got this is like a cripple. You understand? Very serious. That's what we said. Some unlucky people get side effects that are really terrible. So this issue was there in a very small percentage of patients, but for that patient, it is 100% problem. So this is related to PEG, which is considered a very safe molecule for intravenous injection. So there are challenges where people don't understand, and that's why I showed you that picture of the cell, saying that you can run havoc inside the cell. It may not happen in every cell, but it could happen in someone's cell. And this PPES is one such syndrome. This is a study we have done in our lab where we studied doxorubicin, and this is just a plain solution. You can see a huge tumor. And here you can see the tumor has become really small. And this is our active targeting nanoparticles. And here it's not as small, and these are our particles that are not actively targeted. So this is plain solution, untreated. You can see it's really large. This is passive targeting, and this is active targeting. So it is possible to have active targeting. It is possible to have passive targeting. But ultimately, it's all about the technology. This slide is what I've already shown you, so I will go directly here. So when we talk of size-related targeting, it is seen that 200 to 500 accumulates easily in the liver and the spleen. Lungs, if it's even bigger, it goes in. Lymph nodes, tumors, etc. need 100 nanometer or less. So if I need to go here, I will use size of 100 nanometer or less. But now that I have understood stealth, I can go to 200 nanometer and use stealth. So for a technologist, making a 200 nanometer particle with stealth is easier than making a 100 nanometer particle. But we also saw that PEG causes problems. So there's a search for a lot of other stealth agents. So one needs to decide whether 100 nanometer without stealth or 200 with stealth. Very interesting is shape. It is now shown that shape of the particle can also alter the way it goes in the body. I'll directly take you to this graph. They studied discoidal, spherical, hemispherical, cylindrical. 4 shapes. And you can see, let me just take this one liver. In liver, cylindrical is maximum. And then if I take lungs, in lungs, discoidal is maximum. So if I want lung targeting, I may try to make discoidal particles. And if I want mainly liver targeting, I may make cylindrical particles. So this gives me one more opportunity to target a particular organ. And this actually is showing you why shape is affected. When you have spherical, see this coat is forming easily. So it would be recognized by the RES. But if it is like this and it's entrapped this way, this particle would evade RES. But if it is entrapped in this fashion, it would go into RES. So when you have elliptical particles, your biodistribution is not very predictable. But if you have this, it's going to be much more predictable. So there is an advantage, there is a limitation. So we need to actually weigh the pros and the cons. But this is just trying to tell you how shape can be used as one parameter to bypass RES. And I now come to the last example, and this is for active targeting using external stimuli or just stimuli. So what are the stimuli? pH, light, magnetic field. You can also use redox, ultrasound, temperature, ionic strength, use different enzymes, etc. And each of these can give you selective release. The more common for cancer include temperature, pH, ultrasound, and the use of magnetic fields. So here is pH related. One slide which tells you very clearly how at pH 6.8, you're getting much more release of the drug, which does not release at all at 7.4 and 7.2, which is the plasma pH. So it can be in circulation. We saw it's there for 50 to 80 hours, but it will not release. It can release here if it is pH sensitive. Thermoresponsive. Now, when it comes to 39, it will release. And the tumor pH is generally about 39. It's a little higher. Here is an example of NIPAM-based polymers, which have a phase transition at 39.5. And then, what is this? This is an external heating to take the temperatures to 41 to 42 degrees centigrade, which can also be done when the tumor is superficial. So this is the actual physiological tumor pH which can be exploited. We can further take the temperature up and exploit. And again, you find the polymer is NIPAM based, but earlier it was NIPAM with PLGA, which gave that temperature sensitivity, NIPAM with dimethyl acrylamide, which gives this temperature sensitivity. And here is an example for you. At 39.5, just see how much more release. These are fluorescent pictures. And then ultrasound is similar, where when you apply ultrasound, you will see significantly higher uptake by cells compared to absence of ultrasound. The frequency they used was 20 kilohertz, and they applied the ultrasound energy for 30 minutes. And then magnetically triggered targeting, where you can use an external magnet. And what you find here is when I have a magnetic particle, it allows better entry into the cell than a particle that does not have a magnetic particle in it. So here I have the magnet, and when I have a magnetic particle, it enters. So I'm able to enter the cell better. So get better targeting. And in addition to targeting, magnetism has been used for various other options. Like you can also create hyperthermia using magnetism. Drug delivery we saw. MRI is based on the use of magnetism. You can have magnetic biodetection, magnetic separation, lot of applications that you have here. So with this brief, a real kind of overview, I'm coming to this last slide that is optimizing anti-cancer drug delivery. And you may wonder why this carom board. If there is any teacher of physics in this group or any engineer in this group, I'm sure if you play carom or you ever tried to play carom, you'd understand that it is all about trajectories. And understanding trajectories, and it's people who understand that are the ones who are able to actually clean up a board once they have the striker in hand. So that is the challenge. When I hit one coin, if you're a beginner, you don't realize which other coin is going to be hit. You may hit one coin here and another one goes into the pocket. So that is the biggest challenge in anti-cancer delivery. Understanding trajectories, understanding that if I inject this system of this composition, it is likely to go here based on my understanding of physiology. So you need a lot of experience, a lot of understanding of biology, comprehension of the technology, combine the two, and then optimize. So you can't just take the striker and say, okay, I'll keep hitting till something goes into the pocket, because by then you would have caused more damage than actual good treatment to the patient. So optimizing anti-cancer drug delivery continues to be a challenge, although there are many breakthroughs, great strides have already happened, but the challenge continues. I'm showing to you 3 important success stories of anti-cancer nano-DDS. One is paclitaxel, and these are albumin-based polymeric nanoparticles. Then you have doxil and lipodox, both of which are liposomes. And doxil is pegylated liposomes. So stealth liposomes, and I think like this is also lipodox. Interestingly, today, lipodox made by Sun Pharma is the reference listed product in the international market, because the complex technology involved in liposomes, there was a challenge suddenly in doxil, and lipodox came up as the RLD. So on the nano front, the Indian pharma companies are also doing very well. The academicians are certainly putting in their heart into a lot of studies. And we are also working extensively in anti-cancer and treatment of infections. Here is a picture of anti-cancer nano-DDS in clinical trials. It's a very crowded slide, but you can just take one quick look. So it's used for solid tumors, metastatic breast cancer, myeloid leukemia, hepatocellular carcinoma. And here see thermodox. So one of the thermosensitive formulations is also in phase 3 clinical trials. So before I come to the institute that we work in, one very important aspect that our lab is working on in this wonderful institute where I belong, the Institute of Chemical Technology, is, of course, nanomedicine for cancer and infectious diseases, but we are also working on simplifying the technology. Simplifying the technology of preparation of nanoparticles and making it so simple. We have one formulation that has been very successful and it has already been licensed to a company, and we hope that it should be launched in the next 6 months or so. So that is again an important focus that we are working on. And materials, safe materials for nanotechnology and nanomedicine is again a huge area of research that anybody could think of working on. So with this, I would like to just introduce you to this institute that I come from. It's more than 85 years old, ICT Mumbai, centrally located in Matunga. I'm sure all of you know, but for those who don't. And we have since 2018 2 campuses, one in Bhubaneswar and one in Jalna. So we are now 3 campuses. And this is my research group that works very actively in this area of nano. And we've also written 2 books. I've written 2 books on targeted drug delivery because that is one of the major thrust areas that I work on. So with this, thank you very much.
Thank you, ma'am, for wonderful session. You have simplified the things, and that was really great to for the audience like we have for this particular program. Simplifying your presentation to the audience, which is mixed in culture. So thanks for that. And one question, ma'am, you have just given one analogy about the anti-cancer drug, and you have correlated with the carom board, and you mentioned that there is a striker that may be like a medicine for anti-cancer, and you want to hit a coin, and you don't know it may disturb the other coins which you don't want. So the one question pop ups, so that this drugs may cause some side effects when we are developing
Ex-
it for certain thing.
Exactly. So that is what, see, side effects are likely to be there. They are significantly lower when you have targeted medicine. But even with that, I showed you one slide of that hand, right? That syndrome, which is a side effect of liposomal doxorubicin, which is in the market today. So it's not that they're totally devoid and not that they're toxic. The toxicity has come down a lot. For instance, a simple toxicity with anti-cancer medicine was loss of hair. Right? At one time, anybody who went on therapy used to lose their hair. Today, with the monoclonal antibody, that doesn't happen. So, because it is focused for therapy and it doesn't go to the other parts of the body much. So, lot of effort is done. So, what I am trying to project there is when I'm trying to do something new, something different, I need to address it very carefully like this. To see that I just look at this, I want to hit the queen, and I can't just focus on hitting the queen. I have to understand that if I hit the queen, whether when the queen moves, it will go and hit 3-4 other coins of my opponent in carom, which I don't want. So something like that. So there, whether it would go and hit something that would give me toxicity.
Yes, thank you, ma'am, for the nice explanation. So over to the audience again. Any questions if they have? Yeah, there is one compliment for you, madam, from one of the participants in chat box. You can see that.
Yeah, thank you very much. I'd be more happy if you ask me questions rather than just say thank you, thank you.
Yes, participant, please
Because
discuss it. I
ultimately,
mean, that is very
teachers,
important.
because this is a faculty development program, and I think we teachers have to learn to ask questions first. Then only we can motivate our students to start questioning. So it has to begin from us as teachers.
Madam,
So
one
I'm
question from
sure,
me.
yeah, please.
Ma'am, what is the challenges to keep the particle size of the drug at nano level?
Okay. One is it's not the drug alone, it is the drug in the nanocarrier. So there are various methods by which you can develop nanocarriers of small size. I didn't, because of lack of time, I didn't take it. You have, downsizing methods, and you have top-down approach, bottom-up approach, etc. The challenge in keeping it less than 100 nanometer definitely is greater because the high surface area at 100 nanometers, and there is a tendency for them to agglomerate. So that is the biggest challenge.
Okay, I think so. There is no more question. On the behalf of the organizing committee, I would like to thank Dr. Devarajan, madam. Thank you, madam. Thank you so much. You are given a wonderful session. Thank you.
Thank you.
You can leave, ma'am.